because it's early.
Okay, sir. Okay.
Yeah, we'll wait 2-3 minutes. It will be okay.
All right.
Because I think only 57 has joined so far.
Yeah, let's wait for some more time. Good afternoon.
सर ईमेल कहाँ भरनी है?
I think Dr. Rajesh, you can unmute others, no? Because many sounds are coming in between.
Yes, sir. I have muted all.
Okay.
Except all the mic is muted.
Okay. Now it is fine. Now it is fine. Hi, Dr. Mike.
Sir, can we start? Or
Yeah.
Good evening all. So today we are here for the second day of the webinar. So in order to start with the program, I request convener Professor Pranab Chandra Kalida to give an introduction about the speaker and the participants. Please.
Am I audible?
Yes.
Okay. Respected dignitaries of Central Agricultural University, respected Dr. Mike, respected Dean Madam, my colleagues, students, and participants from various institutions of the country. I am extremely overwhelmed to get this opportunity to welcome you all on these international webinars. The first of this kind has already been over on 22nd of July, where Professor Edward Ramsey from University of Tennessee delivered a talk on canine distempers, its praise to the various species, and its prevention. Then CBSE and AHLEC organizes webinars consistently for students and faculty to gain information and to be effectively associated with professional knowledges. As you know, the stem cells have pluripotency to differentiate into many types of cell lineages. Recent progress of avian biotechnology enabled us to analyze the developmental fate of the stem cells. Fertilization of the sex reverse sperm could lead to sexual alteration of the offspring. These strategies using stem cells or the primordial germ cells should be one of the most powerful tools for future poultry breeding. Advancement of stem cells engineering and molecular biology could contribute to avian biology and the poultry industry. Poultry regeneration using embryonic and somatic stem cells, poultry cloning and regeneration of transgenic poultry should be one of the most important subjects. Today's speaker, Dr. Mike McGrew, is a senior lecturer at the Roslin Institute, from where Dolly was born, the first mammal clone from an adult somatic cell in this particular month of July, in the 5th July 1996. Roslin Institute, part of the Royal Vic School of Veterinary Studies at the University of Edinburgh, United Kingdom. Dr. Mike received his PhD from Boston University School of Medicine, USA. He carried out postdoctoral research on chicken embryogenesis at the Developmental Biology Institute of Marseille, France, and on chicken transgenesis at the Roslin Institute. Dr. Mike laboratory studies a type of stem cell in birds, the primordial germ cell, which produces sperm. These cells can be used to generate gene editing sequence which contain precise genetic changes in their genome. He will discuss gene editing and the use of biobanks, that is frozen aviary, with using the germ cells. Avian biobanks will aid in the effort to both manage and conserve both rare and industrial breeds of poultry. Hereby, I invite Dr. Mike McGrew to deliver the talk. Dr. Mike, please.
Thank you. There we go. Can you see my slides?
Yes, sir, it is visible. Yeah, please.
Good, and you can hear me and you can see my my pointer.
Yes, sir, it's visible. Yeah.
Good, thank you. So I want to thank Professor PC for the kind invitation to speak with you. We met 2 years ago at a beautiful, lovely conference in Hyderabad. And actually it was for National Zoo. So it was a very interesting, for me, it was a very interesting meeting because the bird species I work on is the chicken, but I really learned a lot from that meeting, and it was a beautiful visit. And unfortunately, the way things have changed, I haven't been able to travel to back to India for the last 2 years, but I do hope to visit soon. So I'm going to try to give a general talk and aim this for students also. So I know that we probably won't have questions till the end, but please, if you have any basic questions, please feel free to write them down and ask me later on if you don't understand what I'm presenting here, because I intend this to be an informative talk. So I'm going to talk about the research that goes on in my laboratory, and that's basically applying stem cell biology and genome editing to poultry farming. But first, I just want to introduce, here I'm going to try to move this. There we go. Sorry, my screen was. I just want to introduce my university. So I'm at the Roslin Institute. As you heard, the original institute that that cloned Dolly many years ago, and since that time we've moved actually to the University of Edinburgh, and this is our lovely new home here, where we have very large research laboratories. And since there. And since sorry, my screen froze. And since I said I work on poultry, I want to show you our poultry facilities. So we house the National Avian Research Facilities of the UK at the Roslin Institute. And we have two very large poultry facilities. We have where we keep our chickens. We have our standard welfare building, or standard security building called the Greenwood facility, where we house our regular breeds of chicken. And I should say we currently have 15 lines of transgenic chicken and genome edited chicken in our Greenwood building. And we use these to study developmental biology, immunology, and disease resistance. So one of our major interests is identifying genes that are important for disease resistance in chicken for poultry farming. Our second facility is the Bumpsteads facility, and this is what we call our clean facility. So for poultry, this means it's specified pathogen free. So the chickens in this facility aren't actually vaccinated. So we shower in and we shower out, and we have 11 flocks of very special inbred breeds of chickens there where they have specific resistance to different avian pathogens. And we also have been able to introduce genome edited lines into this clean facility. And I'm going on, I'll describe how we do that work. And we supply chicks and eggs to researchers in the UK and abroad. Okay, so as you probably know, chicken is probably the most important and abundant livestock species on the planet. And here I'm showing poultry production. This is back in 2009. And this is showing, sadly, the number of animals that are slaughtered for food each year. As you can say, there's over 1 billion pigs that are killed each year for food. But if you look at chicken, this might be a surprise for you, but there's over 50 billion chickens that are grown on the planet and slaughtered each year. It's a huge number, and this was 12 years ago now, and the number has grown to over 60 billion chickens consumed on the planet each year, and that's 8 chickens per person on the planet. So you can imagine the amount of resources that are going into producing these animals. And one reason, as you know, that chicken is the preferred nutritional meat for feeding the world population is that there are really no restrictions, religious restrictions or social restrictions to consuming this animal compared to different animals, livestock animals. So for poultry farming, we have basically 2 general systems or two general production systems that are in place. So we have commercial farming in very large concentrated facilities, and we also have what we would call backyard farming or village farming, which is very dispersed and also accounts for a lot of the chicken production. Commercial farming really relies on specialized breeds of chicken that have been selected for the past 70 years. They grow very quickly, and they really need a rich, productive, a rich diet to be productive. These chickens are actually genetically diverse in their own way, in between different flocks, and they grow very quickly. But they require a controlled environment to grow, with controlled temperature, controlled lighting, and a high nutrition. We also have what we call local farming, or backyard farming, which mostly relies on indigenous or local poultry breeds. So these poultry breeds are very colorful, very different than commercial poultry breeds. They're locally adapted, we believe. So there's different ecotypes in each country, and they're adapted to local conditions. They have much lower production in terms of weight and in terms of meat and egg production, probably because they're fit, they survive better in the environment, but they actually produce less meat and eggs. But these chickens are actually highly biodiverse. They're very selective for in their environment, and they contain a high degree of genetic diversity that isn't found in these commercial breeds. And they, again, are adapted to low input diet and scavenging feeding. So in my lab, I would say one of our major interest is learning how to biobank, or that's cryopreserve, these different breeds of chickens. And we really want to apply that to indigenous breeds of chickens, but also to commercial breeds. I should say at this point, this is really important because, as I said, 60 billion chicken are produced on the planet each year. Well, none of this comes from cryopreserved flocks of birds, because we don't have the technology to actually freeze down a breed of chicken and bring them back. This is all done from extant living flocks. And this makes us think that the biosecurity of poultry production is very limited.
Recording stopped.
Sorry, are you okay? You stopped
Recording
the
in progress.
Okay.
No problem. You can go ahead.
Okay, sorry, I thought there was a problem. You can hear me all right, yes? Okay, I'll assume that's a yes.
You're audible.
Thank you. So, sorry, as I was saying, we're interested in developing technology to biobank these breeds of chicken, indigenous breeds, because we think they're special, or they have special genetic characteristics, and also commercial breeds. We also want to apply genome editing to actually identify breeds that actually adapt chicken to climates, even warmer climates or tropical climates. And we want to be able to validate these different genes and different traits in indigenous breeds, and either be able to either select these in. Sorry, I can hear voices in the background, but that's okay. Either we can select these traits in indigenous breeds, or we could introduce these into commercial breeds to make them more productive in tropical environments. Okay, so my two big points I'm going to present today is how we are developing avian stem cell technologies to conserve or biobank avian species, and also how we're applying genome editing technologies to bird species. So as you may know, you may have been taught, this is for the students in your reproductive classes, is that reproductive technologies in mammals are very developed. Here I'm showing you a picture of an early zygote. This is a mammalian zygote after fertilization. It's this very large cell, which has just been fertilized and will start to develop. At this point, you can see a needle being inserted into this. So this is actually how Dolly was cloned. We can easily manipulate this structure. We can remove the nucleus from this and put a donor nucleus in. That is how cloning was done. And we can also introduce DNA into this, or our genome editing tools to genetically modify this. So all these technologies are very well developed in mammalian species. We can also freeze down this zygote after it's fertilized, or the early embryo. So we can store mammalian species simply by freezing down the early embryo. So all these technologies have allowed us to develop genome editing and also reproductive technologies for mammalian species. Unfortunately, for bird species, things are completely different. And our technology, we should say, for doing any of these in bird species is probably 20 years or 30 years behind what is done with mammalian species. So both cryopreservation of bird species and also genome editing is very difficult. And that's because of this, just the large size of the zygote. So this is, actually this is a chicken oocyte or zygote, just a few hours after ovulation. And it's many, as you know, it's many centimeters across. It's just, this is a single cell, this is the oocyte, and it contains a single nucleus on the surface here. So just freezing down this structure is impossible because of all the yolk in it. And genetic modification of this is very difficult. So on this pool of yolk, there's one tiny nucleus that's formed. So if you want to genome edit this or clone this, you would have to identify that nucleus and replace it. So it's simply impossible. Also, as you probably know, when the egg is laid, it's very large. So cryopreserving the early embryo is impossible. And another problem is that freezing down semen in chicken and other bird species is possible, but the fertility after you freeze it down is very low. So it means we're really lacking any cryopreservation tools or genome editing tools for avian species. So my lab chose to address these points by trying to use stem cells for genome editing and biobanking. And the stem cells we use are actually very special ones. So these aren't embryonic stem cells that you've probably heard about, that they grow from mammalian embryos, so the pluripotent cells. We actually turn to the stem cells for the germ cell lineage. So these are called primordial germ cells, and these are stem cells in our bodies that only do two things. They only do one of two things. In males, the primordial germ cells make spermatozoa. In females, the primordial germ cells make oocyte. So these are called lineage restriction stem cells for the germ cell lineage. What's interesting about these cells is they're diploid. And what's even more interesting is we don't know how to actually propagate the primordial germ cells from mammalian species, but we can actually, which is astonishing, we can propagate the primordial germ cells from one bird species, and that's the chicken. So just to show you a bit of information about the germ cell lineage, and this is for bird species. It's a little different for humans. So I'm showing you, and this is the early egg here, showing it in the shell, and I'm showing you the early embryo in this chicken egg at early stages of their development. So avian embryos and mammalian embryos are very different. When the egg is laid in the chicken, there's over 60,000 cells present in the early embryo at the laid egg stage. And within this pool of 60,000 cells, the germ cells are already present, and these are the primordial germ cells.
Recording in progress.
And as I said, their only goal is to make sperm or eggs. So in the laid chicken egg, there's already 50 primordial germ cells present. These migrate through the early embryo. They migrate out with the embryo and then they migrate to the forming gonad later on. And this is the same as mammalian species. The primordial germ cells are born in one part of the embryo, and then they migrate to the forming gonad. So on their way to the gonad, they move through the vascular system of the chicken, and there's about 150 of these stem cells present. Later, they colonize the gonad, and at this point, there's over 10,000 cells of these primordial germ cells that are present in the early gonad. So these proliferate for a while in the embryo. In the male, they proliferate for the life of the male. They form the spermatogonial stem cells that form sperm for the life of the male. In females, they undergo meiosis during embryogenesis, and the female is born with their pool of oocytes that will last for their life. So in chickens, you would probably have 100,000 oocytes present at a hatch of the chicken, and these go on to form the eggs. So as I said, this is showing the picture of the primordial germ cells here. They're very interesting cells. They have beautiful large nucleus and a pool of cytoplasm. And again, I'll restate, these are the precursors to
Recording
the gametes.
Stopped.
And we are able to culture them long-term
Recording
for the
in
chicken.
progress.
As I said, you've probably heard of chicken embryonic stem cells from mammals. So the interesting thing is chicken embryonic stem cells, we're able to culture those, but they're not germline competent. They don't make gametes. And we think this is because the germ cell lineage and the stem cells for the body are separated very early in avian development. So the germ cells are set aside early on, and the rest of the cells, the embryonic stem cells that make the body, make everything but the germ cells. And that's different for mammals. For mammalian species, the embryonic stem cells can actually form gametes in the body. And that's because for mammals, the germ cell lineage forms much later in development compared to chicken.
Recording stopped.
So this is showing how we can biobank and culture these primordial germ cells. So we simply take an early chicken embryo, and
Recording
this is just after the embryo has formed. And we can simply, as I said, the primordial germ cells or the germ cell lineage is migrating through the blood. So we can simply sample the blood from a chicken embryo and put these cells into culture. And in my lab, we came up with a defined culture medium that's commercially available to be able to propagate these cells. So in the vascular system, there's only 150 of these cells. But if we put these into culture with the blood cells lyse and the primordial germ cells continue to grow. So within 3 to 4 weeks, there's over 100,000 of these primordial germ cells. So at this point, we can freeze them down in Eppendorfs, put them in the freezer, and thaw them. And freeze them for later use. And this is just a slight video, a short video of showing this process. So this is the early embryo. So this is before the nervous system has formed. So it's a high welfare. It doesn't impinge on the health or the welfare of this embryo. The nervous system hasn't formed. And we simply go in with a capillary and with mouth aspiration, we can go into the early vascular system and take out a microliter of blood. So within this microliter of blood, there's about 10 of these primordial germ cells. So we can simply take this sample of blood and put it into culture. There we go. So this is also a key to how we genome edit and the animals. So we can manipulate these primordial germ cells in culture, or we can freeze them. It doesn't matter either. And then we can take these cells and we can inject them back into a host embryo. So simply we cut again a hole in the shell of this early embryo, and this is before the nervous system formed, and we can simply inject the primordial germ cells back into the vascular system. And they see the host, they can't tell the host from their normal embryo that they're developing in, and they go on their way and they migrate to the gonad and they colonize the gonad and will form sperm or eggs depending on the host. So we simply inject these primordial germ cells from any donor chicken into the surrogate host and raise that to hatch. At a later stage, if we breed from it, some of the offspring, not all, because the host has its own primordial germ cells, some of the offspring will derive from the donor germ cells. And this is showing, in this case, we used a donor germ cell that was GFP positive. It had a transgene that made a green fluorescent protein. So some of the offspring from that surrogate host are green, so it means they came from our donor germ cells. So this is our premise behind how we can biobank chicken or breeds of chicken. If we can culture the primordial germ cells from any breed of chicken, we can put them in the freezer, and later on we can put them back into the surrogate host, and that germ cell will form gametes. So instead of freezing down sperm or eggs, we can freeze down the primordial germ cell. Well, there's a problem for this because the host has all of its own endogenous germ cells. So if we put these donor germ cells back into a surrogate host, very few of the offspring will be derived from our donor germ cells. So the whole system is very inefficient. So our idea, and people have thought of this for mammalian species, is that, well, if your surrogate host doesn't have any endogenous germ cells, all the offspring will derive from your donor germ cells then. So you would have 100% efficiency and you could reestablish breeds. So our idea was if we could use genome editing, we could actually knock out a gene that was important for fertility in the surrogate host. Its germ cells would not develop, and then all the offspring would be derived from our donor germ cells. So I will take a short segue now to talk about genome editing. And to make this perfect or sterile host where all our offspring would be derived from the donor cells, we had to genome edit the surrogate host so it no longer would have germ cells. So as I'm sure you've all heard of, we have this new technology called genome editing or genetic modification, precise genetic modification. And if I can give an analogy, if you imagine that our genome is like the words in a book, and our whole body contains a whole genome in each of these cells, and this genome actually spells out all the proteins that make up our body. And you could imagine this like words in a book. So we've had genetic modification. Well, we've had transgenic technology for many years now, and that would be equivalent to actually adding a extra sentence that was introduced into this. But genome editing is very precise, and that's why we're very excited by this technology. So genome editing, you could imagine, is going into this book that contains all these words that make up our genome and make up ourselves. And this genome editing is so precise, we can go into this whole book of letters that makes ourselves up, and we can change a single letter in one word of that whole book. That's how precise this is. And that's why we're so interested by this technology. So we can go into this and any animal,
in progress.
And this is actually true for humans, potentially for humans, but for any livestock animal or plant. And we can simply delete one of these letters in a single word. Or we can go in and we can change one letter and rewrite it. So that's how precise this technology is. Genome editors basically are molecular scissors. They go in, there's nothing that special about them. They will go in and they will cut at a particular site of DNA. And at that point, you can give it a template, and they will use that template to repair the DNA at that particular spot. So this is how you rewrite the genome at a particular spot in the genome. What happens there is two different things. If the genome editor cuts at that spot in the genome, it can lead to a small deletion. So this leads to a knockout, and this is called the NHEJ pathway. Or if you give it an exogenous template, you can rewrite the genome at that point. This is called precise genome editing, and this uses the homology-directed repair pathway. So both of these are very precise. You can knock out a gene at a particular spot, or you can actually rewrite the genome at that particular spot. So what we did is we used genome editing technology to knock out a gene that was important for fertility in birds. I'm just going to show you the title here. And we used the genome editors called TALENs, and we knocked out a gene that was called DDX4. And DDX4 is important for the germ cells to actually develop. And we knocked this out in chicken. And the idea was what we observed, I'm not going to go into at this point, what we observed is that the females that had this gene knocked out no longer had oocytes, they were sterile, but the males were perfectly fertile. So what we did is we injected female primordial germ cells from a donor breed of chickens into these female embryos that were sterile, and we bred from them. And what we showed is that the offspring, all the offspring came from our donor germ cells. So by using a sterile surrogate, we were able to get 100% transmission and improve our biobanking efficiency. So as you know, people are very concerned about genetic modification of animals and plants, and if these should be used for consumption or not. So the idea is, I won't point out to you if you think genome editing is good for food consumption or not. But the idea was that if we use genome editing to create a sterile surrogate host that could be used for conservation of bird species. And we reported this in the press in the UK, and we got actually very positive feedback. So the idea was that by using genome editing to help conserve other bird species, this was seen as a very positive use and a very positive output for genome editing. So this was in all the newspapers in the UK, and they said this would be very useful. So what I'm going to show you now is I'm going to explain a little bit more on our genome editing technique of primordial germ cells. And I'll show you what we published recently. And this was how we've improved our biobanking technology. Oops. I don't know what that came from. And what it is we decided we realized if we actually produced sterile surrogate host females and sterile surrogate host males, we could streamline this production of biobanking. So if we took our stored, our cryopreserved primordial germ cells, and we injected them into a sterile host bird, we could directly mate the sterile female or the sterile male together and produce pure offspring. So in the example I showed you before, we only had a sterile female host. We didn't have a sterile male. But if we actually had both sterile male and sterile female hosts, we could directly mate them together and reconstitute a breed of chicken. And we called this process surrogate sire, surrogate dam mating. So it's mating your sterile surrogate sires together. So to do this, we produced a transgenic line of chicken through genome editing technology. And these would be inducibly sterile. So you have to be able to breed your surrogate host, and then somehow induce sterility in them, so then you can induce your exogenous germ cells. So what we did, this is a bit of complex molecular biology. But what we did is we used genome editing technology, again with a CRISPR, to nick the genome, to cut the genome in a gene that was only expressed in the germ cells. And the gene that we chose as our host locus is called DAZL. So the DAZL gene is only expressed in the primordial germ cells, in the germ cells of the animal. So we designed a CRISPR-Cas9 to cut the C DAZL locus at a particular spot in the genome. And then what we did is we recombined a gene into this locus called iCaspase9 and also a GFP reporter. So we were able to recombine a reporter gene in the iCaspase9, in the DAZL locus of this particular location in a primordial germ cell. And to point out, I don't know what that circle is on this sign, but I'll just ignore it. I think I gave a talk and I was drawing on these lines. So what the iCaspase9 gene is, to make it very simple, is you might have heard about the caspase9 genes. They induce apoptosis in a cell. So when the caspase9 genes are expressed in a cell, two of the caspase9 proteins come together, they dimerize, and they cause the cell to apoptose. So what someone did many years ago is they took an artificial dimerization domain and fused it onto the caspase9 protein. And this is called the iCaspase9 gene, or iCaspase9 protein. Simply what you do is, if this is introduced into a cell, you can add a chemical compound which has no effect in the cell, except it binds to this dimerization domain. And when your cell contains an iCaspase9 protein, the dimerization domain comes together and it induces apoptosis. So what we did is we genome edited a primordial germ cell to contain this iCaspase9 gene that was in the DAZL locus. So this caspase9 gene is only expressed in the primordial germ cells of the animal. So this would be in our surrogate host animal. So what would happen is that, and these animals are our surrogate host. So they're genome edited, but they're also transgenic. So they use both technologies. And as I said, what we did is we produced this line of transgenic chickens that contain this iCaspase9 gene that's only expressed
Recording stopped.
in the primordial germ cells. Oops. So this is showing one of the chickens
Recording
here.
in progress.
And what I'm showing you is this is the early embryo, and this is the embryonic gonad here. So this is the mesonephris. This is autofluorescence in the mesonephris here. So these are the embryonic kidneys, and this is the embryonic gonad here. And you can see this expression. This is the GFP expression because we also put it in a transgene that's expressed with the iCaspase9 in the germ cells. So what we're seeing here is the endogenous primordial germ cells after they colonize the gonad. So this is the iCaspase9 chicken. We add this drug, as I said, this simple compound that activates the iCaspase9 gene. And this is showing the iCaspase9 gene after we add this drug. And what you can see is all the endogenous germ cells are no longer present. So the germ cells are lacking. So this embryo actually is our perfect sterile surrogate host. So if we introduce exogenous germ cells into this, all the offspring would come from our exogenous germ cells. And this is showing
Recording stopped. Recording in progress.
This is showing the whole pathway here. So I'm sorry. So what we do in terms of what we call our new biobanking technology is we can take any breed of chickens and we can isolate the embryonic blood from any breed of chickens. And we simply culture the germ cells to increase their number, and we can cryopreserve them at that stage. So this is our frozen biobank. At a later point, we can take these frozen primordial germ cells, we inject them with this compound called BB, and we inject them back into our iCaspase-9 surrogate host. So that surrogate host then will lack, because we added the compound, it will lack all its primordial germ cells and only produce offspring from our frozen germ cells. We can then breed our surrogate host together to produce a pure breed of chickens. And you can see it sounds very complicated. You know, we have sterile hosts, we have frozen cells, but it actually works really well. And we haven't, I should say, we haven't published this data that I'm showing you here, but this is showing actually how powerful this technology. So this hen on the left and this cockerel or rooster are the surrogate hosts. So these are the iCaspase-9 surrogate hosts. But when they were embryos, we injected them with frozen primordial germ cells that came from a breed of chicken that have black skin. So these were a silky breed of chicken. So these two chickens grew up, we mated them together, and these are their offspring. So their offspring, all their offspring come from a completely different breed of chicken. So this is our surrogate dam mating, and this shows how we can cryopreserve a breed of chicken and reestablish it in a single step. And just to show you more, these are the chickens after they grew up. So again, on the left, these are the parents here. So they're a brown layer breed of chickens, so they're very fertile. And these are their offspring here. And these are a silky broiler breed of chickens. So they're a completely different breed of chicken. They're also broiler versus layer. And these are the offspring here. So a very powerful technology. I'm going to show you one example, and I do believe I have 15 more minutes, and you can tell me if that's wrong. Is that correct?
Yeah, it's correct. Yeah.
Okay, thank you. I want to make sure I'm on time. I'll show you actually how, so that was basically showing how we use sterile surrogate sires for biobanking, but I want to show you also how the system is really important or really useful for producing chickens that are genome edited. So as I said, we have a sterile surrogate host. And we have our primordial germ cells from any breed of chicken which we can genome edit. We published this actually in Nature Communications last month. And what we wanted to do is we wanted to edit a gene that was important for feather color. And one of the genes, you probably know this because you're out of vet school, one of the genes that's important for feather color is called dominant white. So dominant white is dominant because if you're heterozygous for this allele, you will have white feathers. So this gene is actually very important in broiler chicken production because they like to have white feathers. So most broiler chickens will contain the dominant white locus. So what dominant white is this a natural occurring mutation that was found in chickens, and leads to a mutation of a gene called PMEL17, which is important for melanin production in the feathers. So dominant white chicken have a mutation in this gene, so they no longer make black pigment. So this dominant white allele, actually, this was mapped by my colleagues in Sweden. This was mapped by Leif Andersson's gene group, actually. And they found that dominant white was actually a 9 nucleotide insert into the PMEL17 gene, which leads to an insert of a W, A, and P amino acids. And this led to a dominant loss of function of the color allele. So we used our genome editing tools, and we used primordial germ cells from a dominant white breed of chicken, and we simply transfected our genome editing tools into these primordial germ cells. And this shows,
Recording stopped.
in our single trace here, it shows that we were able to genome edit the primordial germ cells and change the genetic
Recording in
locus
progress.
and remove the dominant white mutations from the primordial germ cells. What we simply did then is we injected these primordial germ cells back into our sterile surrogate host. We edited male primordial germ cells and female primordial germ cells at this location and injected them back into our sterile surrogate hosts. So this shows this breed of chickens before we genome edited, and this shows that the chicks they're dominant white, they have a white feather color, and when they grow up, they have these white feathers, so they're perfectly white. So from our genome edited primordial germ cells that we took from this breed of chicken that we put into our surrogate host, this was the offspring that were produced. So it was amazing. All the offspring, now the chicks had black feathers, and when they grew up, we had no idea what actually colored chickens these would be, but they turned out to have, the females had barred feathers, and this is showing two genes here, and these, the males had actually speckled feathers. So in this chicken that was dominant white, hiding underneath the dominant white allele was a different feather color allele, and this was to make barred feathers. So this is showing actually the power of this technology. So in the final few minutes here, I'd like to show you how we're improving our biobanking technology. Now, as I said before, we take any breed of chickens, we have to get fertile eggs from that breed of chicken, and we have to isolate the embryonic blood at an early stage, and we have to culture them in vitro. So it's very labor intensive, and it's very high. It has a very high technology. So we have to be able to go and sample chicken eggs. We have to be able to culture the germ cells, and then we can freeze them down.
Recording stopped.
So we've been working with a laboratory.
Recording in progress.
In ILRI, this is the International Livestock Research Institute, to bring this biobanking technology into Africa to help preserve the breeds there. And this is through the Center for Tropical Livestock Genetics and Health and through funding from the Bill and Melinda Gates Foundation. So they've actually been able to culture primordial germ cells from 7 different ecotypes or local breeds of chicken in Tanzania, in Ethiopia, and in Kenya. So they have frozen down 15 different breeds and over 400 different genotypes. Well, as I said, this is fine. This is very stored, but it's very technically difficult to do this work. So, as I said, now that we have this sterile surrogate host, we should actually be able to develop this technology and not have to culture the primordial germ cells before we freeze them down. Because as I said before, culturing these primordial germ cells is very technically difficult. But the early embryo, actually, as it developed, the early embryonic gonad contains several tens of thousands of primordial germ cells. So if we are able to biobank this early material, embryonic material, which contains tens of thousands of cells, we shouldn't have to culture the primordial germ cells. I should mention, this work is done with funding by UK Aid, which is a UK aid agency for overseas development. So simply what we do is we take the early embryonic gonad after the germ cells are migrated to the gonad, and we can cryopreserve those. And this is how we can biobank instead of culturing germ cells. We have biobanked this material, and later on we simply re-inject it back into the surrogate host. And what's interesting for the germ cells in chicken is that the primordial germ cells are able to re-migrate back to the gonad if they're injected back into the blood. So they're completely plastic. So even though they migrate from the blood to the forming gonad, if they're isolated from the gonad and reintroduced into the early blood system, they're able to re-migrate to the gonad and efficiently colonize the gonad. So I should say at Roslin, we have these fluorescent lines of chicken. So it's easy for us to follow the donor germ cells using our chickens that actually are marked with a red gene or a green gene. So in this case, we took primordial germ cells that are present from red or green gonads, froze this material down and biobanked it. And then we injected it back into a surrogate host. And what we saw when we mated these surrogate hosts together, that the offspring from these gonads came from our donor material. So what we found is, and I would say, we believe all the offspring came from our donor gonads that were red or green. So in this case, we got yellow offspring. But what this shows us is that we are simply able to cryopreserve the embryonic gonads from early embryos to be able to preserve a breed of chicken. And then we introduce these into our sterile host, breed them together, and produce pure offspring. So I will finish my talk at this point. So what I've shown you, I hope, is, sorry for this little circle here, but what I've shown you is using primordial germ cells, we're very able to efficiently genome edit chickens.
Recording stopped.
We've produced a sterile surrogate host that we're able
Recording
to
in
introduce. We can introduce donor germ cells into it. And we believe this actually is a welfare-friendly system. I didn't mention, but the only other way to make a sterile surrogate host is to treat with chemical compounds to try to destroy the endogenous germ cells. So these are chemotherapeutic reagents, and they're very harsh on the surrogate host. So in our case, the only difference for the surrogate host is that it's sterile. It has no other welfare impacts. And by using this system, we can actually, by introducing male and female donor germ cells, we can re-establish a chicken breed within 6 months. And we think this technology will be very useful for biobanking chicken breeds. So the problem is, I've shown you our results for chicken. And, that's one bird species, and there's over 10,000 bird species on the planet. And that's something we can discuss, but because so far we've only been able to adapt this technology to the chicken. But wouldn't it be amazing if we were able to do something for some of the different 10,000 bird species on the planet that we're interested in? Oop. And just to show my funders in this one slide, and all the people that are involved in this work for helping me in this, or actually who accomplished this group, from my group, from the Roslin, and the other institutes also in other countries. And I'll stop sharing my slides there, and I'm very willing to take any questions.
Is there anyone who is unmute? Please mute it.
Dr. Rajesh?
Yeah.
Yeah, I think.
Thank you, sir, for the presentation. So now is the time for question. I feel two questions. are already there, and I would like to read it one by one.
Okay.
Where is the first one? Yeah, the first one is generation of avian influenza resistant chickens through genetic modification and or selective breeding may help prevent viral spread, creating disease resistant chickens, a viable solution to avian influenza. So what's your opinion in this?
I would hope so. I mean, that's it. The problem is that genome edits to increase resistance or to validate. There's two ways to look at this. We can use genome editing to validate alleles or genetic changes to show that they're useful for disease resistance. If these alleles or these genetic changes don't exist, we could try to artificially create a genetic change that would make the animal resistant to different viruses. And I think it's worthwhile doing for some of the viruses that we have no vaccines for in poultry livestock. So for avian influenza, it would be very important to be able to do this, to do it. Unfortunately, it's incredibly difficult to genome edit for genome edit for resistant to avian influenza. We do have one target, and we've shown this online. There's a gene called ANP32A, and we believe by genome editing that gene, making a subtle change by 2 amino acids in that gene, the chicken will be resistant to avian influenza. Resilient or resistant. So we're investigating that. And if we can prove it's true, that the chicken is completely resistant, but it's more difficult than that. The chicken can not only be resistant, it can't be a carrier of avian influenza. So that's the other problem. So to generate disease resistance, but not actually make an animal that will carry and spread the disease. So if so, yes, that would be very useful.
Okay, thank you, sir. Now, the next question is, for cryopreserving the stem cells, is there any standard protocol?
Actually, no, because we're making that up as we go. So we're just publishing, and it's, as I said, we've developed this technology. We've published that. We haven't even published that we can bring a breed back from the primordial germ cells. We haven't published what I've showed you yet with those cultured germ cells and the chicken with the black skin, using the frozen gonads. We've just finished that work, so we haven't published it yet. We think in my lab that we have a standard protocol. Yeah, so we're trying to write it out and validate it, and we do want to share that with different research laboratories. Exactly.
Recording stopped.
Especially around the planet. So we hope that
Recording
this technology can be adopted. But I should say, short answer is we have a written protocol that we can share out, but the technology isn't proven yet.
The next question is, what was the status of F2 and other generation of chickens, especially in the silky chicken example? Is there any effect of crossing over on the knockout gene, or is it possibly it can revert back to wild type in future generations?
Yeah, so the important thing is the surrogate host doesn't transmit any of its genome to the offspring. So not mitochondria, not anything. So it's only a host to the germ cells. We've looked at the genome of the donor cells. So that was the silky. So none of the offspring in that experiment came from the ICAS-9 surrogate host. So as far as we can tell, they're a pure offspring. The problem is, when we culture the primordial germ cells, we culture them from a single embryo. So when we had huge numbers of primordial germ cells, hundreds of thousands from a single male, and also hundreds of thousands from a single female. And we injected those back into the surrogate host and we crossed them together. So what we did is we produced huge numbers of all those embryos shown there, even though we had multiple surrogate hosts, many males and many females. All the offspring were full siblings, brothers and sisters. So they were all genetically related. So that was our big problem. So you might know for poultry farming, you want to have a lot of genetic diversity in your offspring. And we actually, using our surrogate host, they were full siblings, so they were perfectly healthy. But if we mate them on, they're going to become inbred for our next generation, our F2 and our F3. So we didn't realize till afterwards that actually we have to be able to biobank multiple genomes, because we're not allowed to breed on full sibling offspring because they're too related. So yes, F2 generation would be healthy and F3 generation would be healthy, but that led us to say that we have to be, how we biobank, we need to change, we need to freeze down multiple genotypes. I hope that answered part of your question.
Recording stopped.
Yeah. Thank you, sir. So now I feel...
Recording in progress.
Yeah, one more question has come. So please give advice on whether it is safe to consume genetically modified species by humans.
Oh, that would be... Oh, thank you. I was going to say. For me, my personal experience, or for the world. So that, are you talking about, sorry, was the question genetically modified or genome edited? Because that's a different question. I mean, that's transgenic versus genome edited, and different amount, some people would say different amount of risk. And since you're recording this, was the question, is it safe? Is the question, is it safe to eat a genome edited plant or animal? If that was the question, for plants, it's very interesting. So for plants now, if you look at how plants are selected, a lot of times, or in the past 50 years, they've gone in and to generate new varieties of plant, they've gone in and irradiated the plant species to induce random mutations in the genome of the plant, and then they bred on through that. So plants have already been genetically engineered through natural tools, not through genetic tools. And that whole process is very inaccurate. The genome has been randomly mutated throughout the whole organism. So genetic engineering tools are, to me, they're a lot better and a lot more precise than what was used in the past. So that's in terms of plants. So that's changing a single base pair. People say, oh, there's off-target effects. There's off-target effects that, the genome editing tools can cleave randomly in the genome. Well, there's off-target effects when you have children, because each time you go through a generation, you have 10, for humans, 10 to 30 random mutations in the genome per generation. Off-target effects are part of our life, so that's not a big deal. So in terms of genome editing, it's actually very safe, because you're not introducing a transgene, and you're not
Recording stopped.
You're not introducing any new allergens in the animal.
Recording in progress.
So to me, I would say in terms of plant breeding, there's not a problem there. And that's true for animals also. If it's transgenic, it depends on the transgene. If it's shown that it doesn't contain an allergen, then it should be safe for humans. So the technology itself has no value. It's neither safe, good, or bad. It's how we use it, and it depends on the end product. I hope that was more
thank you,
precise.
sir.
You're welcome.
Yeah. the one who was asking about the genetic edited plants and animals. I feel this answer.
Yeah.
Okay, thank you, sir. I feel these are the questions I could see in the chat session.
Okay.
So now I request the participants. If anyone would like to
Recording
ask or
stopped.
interact with the speaker, you may kindly unmute your mic and you can speak, please.
Recording in progress.
Good afternoon, sir. Good afternoon, sir. This is Dr. Bimal Chaudhary from Odisha.
Yeah, audible. Please go on.
My question.
Yeah, please proceed.
My question is that sir, Dr. Mike told that there is a MP32 gene which can be modified, and that will actually check the avian influenza. But still, the bird will be the resistant, and also will be the carrier. So, can you just throw some more light on it?
All I can say at this point is that we have a project, because unfortunately we did a press release, but the gene, it's been published that the edit of this gene
Recording stopped.
led to resistance from infection in cells.
Recording in progress.
So they was shown that by, and we showed that too, by editing cells in vitro, that it led to resistance in the cells. And we have, I should say, unfortunately, we have funding from the UK government, and which was released, and from a company to actually test this in chickens. So we're currently trying to test this in chickens to show and then to produce genome edited chickens, and then to challenge them with avian influenza. And then we have to find out if that's true. Are the chickens resistant? Or are they completely resistant and not carriers? So that all needs to be tested. And then if that proves to be true, then the next steps would be, we would have to think about if this would be actually useful to introduce into poultry populations. So these sort of edits, the thing is, genome editing can validate. What would be really lovely, as I said, we described the gene ANP32A. If we could find natural variants that exist out there for the chicken population, we could breed from them, and that would be a resistant chicken. And it would be, in terms of how everyone seems to be afraid of genome editing and all the regulation, if you could find an animal that contains that natural change somewhere in its genome, and I said there's 60 billion chickens out there, there's going to be a lot of variation. Why not just select for that animal and breed from that? So in terms of, I can't talk about the ANP work because unfortunately that work's not published. We're still working on it, but the idea is we hope that would go forward.
Thank you.
Okay.
Thank you. So anyone else who would like to interact with the speaker?
Myself, sir, Dr. Mansi Shukla. First of all, a very good evening to the very respected speaker, sir, from Rewa, Madhya Pradesh, India. Myself, Dr. Mansi Shukla, in anatomy department here, sir. Sir, my question is, though it may sound very simple, but I was wondering, sir, are there any chances of immune rejection from the host cell in which the primordial germ cells are inoculated?
That's an excellent question, because we were very much afraid of that, because we were afraid that the endogenous germ cells could be rejected. What's interesting usually is self-tolerance, because we put the germ cells go in at an early
recording stopped.
The germ cells go in at an early embryonic stage.
Recording in progress.
The animal, when it's introduced at such an early embryonic stage, the animal will learn to self-tolerate. It will see the germ cells as self. So when they develop, they shouldn't be rejected, at least for the male. For the female, we were worried about that. And what our current worry, because we worry a lot about this, how's this technology going to
Exactly,
but
sir.
what we're interested in is mixing multiple genotypes in our surrogate host. Because I said, when you bring back up a breed of poultry, you need to bring back hundreds of different genotypes. So that's what I said. That's one of the reasons why we simply switched over to freezing down the gonadal germ cells, mixing them together.
Recording in progress.
Our new fear is that some genotypes might be rejected. And that's what we're currently measuring. If we take germ cells from 10 different individuals and put them into the surrogate host, will we get offspring from all 10 different genotypes? And but potentially some of the genotypes could be rejected. And also the other problem is there's sperm competition between germ cells, which is such an interesting thought. So what happens when we have a surrogate host that carries 10 different genotypes? What's going to happen with sperm competition for the female? Because we think the female might choose which semen it wants to be used to inseminate the oocyte. So what is the female going to choose some genotypes to transmit?
Recording in progress.
So it's a very good question. Thank you. And there's a lot of questions we don't have answers for.
Ji sir, definitely. Thank you so much, sir. But this sounds interesting, sir, because you were saying that now these germ cells would be considered as cells.
Yes.
This is an advantage, and it could be applied to mammalian species also if more research is done in this context. Mammalian species germ cells are taken and introduced at that stage only, primordial germ cell stage.
Well, so they're doing so my colleague in University of Washington, they made a sterile They made a sterile surrogate goat, and
recording in progress.
And they're trying to transplant spermatogonial stem cells into the sterile surrogate goat. But again, this has to be done in the adult goat. They can be easily transplanted in, because in mammals, these experiments are very difficult to do in mammals. You have to isolate the embryo. You would inject them into the embryo, and you would have to implant into your surrogate host, dam or further.
Yes, sir. Though it is difficult, but it would be of great advantage because the cells would be considered as cells, and what we fear is of immune rejection. You are talking that there is rejection because of different genotypes introduced.
Recording in progress.
That won't be there if we consider this primordial germ cell introduction. If there would be type, but we consider this fact that we take these primordial germ cells, then it would be advantageous, and it would be very advantageous rather, because immune rejection would be least.
You're right, because a lot in the adult mouse and in the adult goat, actually, I don't know if they did this for the goat, they treat with busulfan to eliminate the endogenous cells, and actually I think to help, and also they treat sometimes with drugs to suppress cyclosporin to suppress the endogenous endogenous immunity, so they aren't rejected.
Recording stopped.
You're right, exactly. So using primordial germ cells might be helpful. Yep.
Recording in progress.
Thank you so much. It was an interesting.
Yeah, thank you.
Okay, thank you, sir. I feel these are the questions and concerns. So now I request Professor Pranab Chandra Kalida to propose a vote of thanks. Please, sir.
Thank you, Dr. Rajesh. So it was really great that these international webinars was attended by 153 participants. And at present, I am here to present the vote of thanks to the people who made this event successful. I would like to take a minute and thank all of you. First of all, I would like to thank our webinar speaker, Dr. MiKe for enlightening us with his knowledge. Today's webinar was full of knowledge and interesting things.
Recording
which is
stopped.
explanatory from the various interesting
Recording in
questions.
progress.
It gave deep insight into the topic and also revealed some of the interesting facts, especially the genome editors and the positive stories on conserving using sterile host chicken was really interesting. I am pretty sure the precious knowledge that Dr. MiKe gave us will definitely help our colleagues.
PC Kalida sir, please unmute.
Sorry, I'll start, yeah. It is really great that this international webinar was attended by
Recording stopped.
150 participants. Now I am here to present the vote of thanks.
Recording in
to the
progress.
people who made this event successful. I would like to take a minute and thank all of them. First of all, I would like to thank our webinar speaker, Dr. MiKe McGrew, for enlightening us with his knowledge. Today's webinar was full of knowledge and interesting things, which was self-explanatory from the various interesting questions. It gave deep insight into the topic and also revealed some interesting facts, especially the genome editors and the positive stories on conservation using sterile host chicken was
Recording
really
stopped.
interesting.
Recording in progress.
I am pretty sure the precious knowledge that Dr. MiKe gave us will definitely help our colleagues, students, and participants in their studies and future career. Once again, I would like to thank Dr. MiKe McGrew for taking out time from his busy schedule and enlightening us with his knowledge. Dr. MiKe is very busy in delivering talks in various international forums pertaining to his laboratory work. Thank you so much, Dr. MiKe. I would also like to thank our Dean Madam, Professor Mar, for giving permissions to organize these international webinars and inviting Dr. MiKe to deliver the talks. I want to thank Nodal Officer Dr. Ali, Dr. J.B. Rajesh, Member Secretary, and other coordinators, Dr. Kulkarni, Nancy, Abhishek, the coordinators for these webinars. Special thanks to Dr. C. Bhanlal Puja, Research Associate, who really worked hard to make this event successful. I'd also like to thank all the teachers for encouraging students to participate and also guiding the participants too. Thank you, all the faculty, students, and other participants from various institutions of the country present here for paying your attention and learning. I end my speech here. Thank you, all of you. Have a wonderful evening.
Thank you, sir. Now, I feel we come to the end of this seminar. Once again, I thank each and everyone. Thank you all.
Hi, Mike. Thank you.
I'll unmute. Thank you. That was great.
Yeah,
I enjoyed it.
I'll in touch. I will contact in touch.
Okay, thank you. And if anyone has questions, they can write to me.
Yeah.
Yep,
Sure,
good.
sure.
Good.
Yeah. Have a wonderful evening.
Thank you.
Recording stopped.